morning, viewer. Welcome to our Glory Globe TV. Here's breaking news. The chains of amino acids that make up proteins are critical to every form of life. The complex ways that these proteins fold and interact has fascinated researchers for decades. Exactly how a protein folds determines its function. For instance, a particular protein can take on the job of transporting molecules, attacking invading cells, or repairing DNA. A significant amount of progress has been made in this field of research, but much is still unknown about the folding processes. Now, all researchers have figured out how to create computer models that simply but accurately represent these proteins, and the results are published in Physical Review E. Viewers, every protein has a specific function, and when it does not fold how to and it should, then disease and other serious disorders can result. A better understanding of how proteins fold could boost the creation of new pharmaceuticals to treat misfolding disorder. So, for the same, current computational models that operate at atomic resolutions, however, cannot simulate the folding of most proteins. The complexity makes the calculations prohibitively long. So, the Protein Data Bank, a public database of protein structures, has the known structures of only 40% of human proteins. Therefore, researchers expressed that we currently do not know the true structures and folding pathways of 60% of protein, said Corey O'Hearn, professor of mechanical engineering, who led the study. The researchers set out to develop a computational model that captured as simply as possible the most important structural properties of folded proteins. Where computational models can have different resolutions, O'Hearn explained, they can describe the atomic scale, that is, they can model every atom in the protein, or the models can be coarse grained, in which groups of atoms in the protein can be represented by a single unit. For instance, the researchers might use a single spherical object to model the backbone atoms of a given amino acid, and another spherical object can represent the side chain atoms of the amino acid. We studied a range of computational models from atomistic to very coarse grain to determine the coarse resolution that was able to accurately capture the structural properties of folded proteins, he said. The researchers compared the predictions of computational models to the protein core density and other structural features from thousands of proteins. They can create coarse grained computational models with simplified representation. Behind this exclusive report, Glory Globe TV, Delhi, Bharat.